the function of an healthy immune system is to taking care of the entire body. So all the diseases that appear with age, they are somehow connected with immune dysfunction. So we believe that by rejuvenating the immune system, we can offer a new solution for a plethora of aging-related diseases.
Sure. Welcome, everybody. This is Buck Joffrey with the Longevity Roadmap, and today, very interesting show, and it is on the topic of, the aging immune system. My guest, Alessio Lanna, has some really fantastic research coming on. He's a founder of a company called Centcell, that's Centcell, which you may want to look up. The interview itself gets pretty technical, so I wanted to kind of give you an overview to help you understand, some of the terminology and what's going on. But effectively, at a high level, the concept is that the immune system, obviously, it plays a major role in aging. I mean, we've talked so much about, in previous shows about mitochondria, but I think another major, thing here is immunosenescence. And you heard Dr. Greg Fahey talk about the importance of the thymus gland in the past and that kind of thing, which goes to that as well. So I do think that the immune system is a major part. And if you look in some of a lot of the diseases of aging, they involve chronic inflammation, right? It's effectively the immune system attacking our own body at a low level, a low grade, and creating havoc. And that is a major contributor to metabolic disease, pretty much all disease, cardiovascular, neurologic diseases. And so, so again, the importance of the immune system and aging of the immune system cannot be understated. But so, at a high level, what really we think of when we think about aging of the immune system is called immunosenescence. So we've talked about senescent cells before. These senescent cells are essentially cells that, normally cells will replicate over time for a period of time, and then they'll go through different cellular phases, and eventually they die, a process called apoptosis. Well, when a cell is senescence, it kind of stops at a phase and it sort of just becomes a zombie. It doesn't die. And that's not good, because those cells not only are no longer functional, but they often release various toxic chemicals called cytokines and that kind of thing that can turn others into zombie cells as well. So T cells are one of the major players in the immune system. And what this research is about, and what this conversation is about, is trying to combat immunosenescence. So, senescent immune cells. The concept is that a lot of T cells go into senescence because the role of telomeres are sort of the caps at the end of chromosomes. As they replicate, they become shorter and shorter, and that's a sign of aging. Now, there's an enzyme called telomerase that helps to lengthen telomeres, but sometimes, that's just not enough. And that's where a concept comes in called intercellular telomere transfer. Okay. This is one of the things that our guest, their group has discovered, where basically T cells can regain these longer telomeres by a process called intercellular telomere transfer. And what they found is that another type of immune cell, a shorter-lived immune cell called an antigen-presenting cell, can actually donate tiny particles, packaged up particles that contain segments of telomeric DNA, and they can transfer them to the T cells. So they become effectively like these donors of the raw materials for the telomeres, and that, of course, is a big find. I mean, why would they do that? Well, because the antigen presenting cells don't live as long. The T cells live for years, and you want those T cells functioning. So that's one of the discoveries early on that was really interesting. Another thing that this group found was that there's a complex called P38 MAP kinase, and also a group of proteins called sestrins. And when they combine, they can actually push these T cells towards senescence, and that typically happens with aging, with chronic inflammation, any kind of stressors, etc. And what this group is found, and this is the big thing here, is they've developed these various compounds called disruptors of sestrin MAP kinase complex. And effectively what they're seeing, not only in cells in the labs, what we say in vitro, but also in vivo, which is in animals, that they are able to use these compounds literally to reverse these immunosenescent T cells. And that is a huge, huge find. The implications are very, very big. And as you can imagine, it's if you are able to, effectively reverse an aging immune system in a human, that has tremendous therapeutic potential for not only health span, but lifespan. So it's a really exciting, really exciting area of research. And, I wanted to make sure I gave you some sort of place to start, because again, the interview itself gets complicated. So if you understand kind of what I said so far as a framework, you'll get more out of it. And, but suffice it to say, it is important. And we're going to have that interview right after these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to the show, everyone. Today, my guest on Longevity Roadmap is Dr. Professor Alessio Lanna. He's a distinguished immunologist and researcher. Professor Lanna holds a PhD in immunology and has served as a faculty member at leading institutions, including University College London. He's the founder of SenCell and is internationally recognized for his groundbreaking work on immune system rejuvenation and T cell biology. Welcome, Professor Lanna. How are you?
I'm very well. Thank you for inviting me.
Yeah, interesting stuff, looking over your research. I want to, before we get into the specifics, just make sure that we give the audience a little bit of background. You're an immunologist, and at a high level, perhaps you could describe for us what happens to our immune system as we age.
Yes, that's an amazing question. So our immune system, first of all, serves as a barrel, like a defense barrel against all sort of insults that occurs over a lifetime. That be infections of various natures, that be cancers and so on. And we're moving towards the awareness nowadays that if we keep an healthy immune system, it's the entire body to benefit, not just the immune regulation, not just immune control. So in other words, we need a good immune response, a cold immune response for our health span. And also lifespan. Some work from my laboratory is showing that affecting the lifespan of the T cells affects the systemic lifespan. We can talk about that later on. But as we age, unfortunately, progressively we lose the ability of our immune cells, particularly our T cells, but actually all the immune cells to do their job, right? So to control infections and so on. So this process is called in the field immune senescence. And this is the process that I've been studying for all my career. The process of all the signaling pathways, things that go wrong as we get older, and how we can get back. I will get better pharmacologically.
Yeah. I want to talk, just in terms of the aging process itself at a higher level first. I had Professor Greg Fahey on the program. I don't know if are you familiar with this work on the thymus? Yeah.
Yes,
So,
some work on the timeless.
yeah. So we had Dr. Fahey on the show, and he believes that, the aging immune system ultimately is because of a shrinking thymus gland as we age. What are
Well,
your
there is
thoughts
simultaneous
on?
involution. Well, yes, there is
Yeah.
simultaneous involution, of course. Once we go older, there is simultaneous involution, and so meaning that the output of newly born T cells is actually reduced. But bear in mind that even if you look in old people, the quantity and the production of the T cells, surprisingly, is not that affected, right? Actually, more importantly, is the ability to keep the cells in the periphery in a healthy state, such that the naive T cell clones will respond in the correct way, and because also of the accumulation of the senescent populations of T cells. There's been work trying to rejuvenate the thymus. Unfortunately, the effect in a variety of ways, but unfortunately, the effect has been temporary. And so data have shown that it probably is more productive to focus on the periphery, because again, the output is not that compromised in terms of naive T cells. So we have naive T cells as we go older. They just don't work very nicely.
So let's focus on the T cells, and I know we've talked about it specifically, but just for the audience, what role specifically do the T cells play in the immune system? So we can sort of go
Yeah.
from there.
Well, you have two main types, obviously, right? CD8s and CD4 T cells. So if you go to immunology books, you will see that the CD8 T cells are important for controlling and killing immediately the infected cells. And CD4 T cells actually orchestrate the overall immune response, they orchestrate B cell functions and so on. But actually, this is a bit more complex than that. And CD4 in particular, they are essential for, again, the overall health span of the body.
When you talk about T cell senescence,
Yeah.
talk about what exactly is happening to these T cells at the cellular level and the role of telomeres.
So they lose the ability to proliferate. So the first hallmark of these T cells is that when they recognize an antigen is a foreign substance towards which the T cells react in order to protect us, right? So they lose progressively the ability to recognize the antigens and thereby to divide, which is a process called clone expansion. So progressively they lose the ability to proliferate and to divide, so to mount a robust immune response. We study these T cells from the blood, so you can get these T cells from circulation with specific loss of markers and acquisition of other markers that allow us in the lab to sort them out and study them extracellularly and inside the cell. And the cells are very important because my work, what we have studied, what we have discovered, is that actually there is a very large complex of intramolecular proteins, which we have called the Sestri-McManus complex, ESMAC, which was published almost 10 years ago now. So this is a gigantic complex that sits in the proximity of the T cell nuclei and orchestrates the overall immune senescence response. So if you target pharmacologically this complex, you can turn back the clock, you can rejuvenate the immune response.
Before we get to the sestrins and stuff, though, in terms of if you could talk about like sort of what's happening with T cell senescence, I understand that one of the key elements there is really telomere shortening. Can you expand about
there is telomere shortening? There is telomere shortening. However, we have to be careful because telomere shortening per se is not essentially a hallmark of of senescence. In other words, there are senescent T cells that do not have very short telomeres. These are called in the field EMRA T cells. So work has shown that senescent T cells essentially comprise two different subtype of cells: effector memory cells and effector memory cells that express a marker called CD45RA. So we look at telomeres in these two senescent T cell populations. Effector memory T cells have very short telomeres. Not so true actually in the T-EMRA cells. So if you just go by telomere length, we lose actually a good proportion of cells that are senescent. So telomere shortening is a feature of some immune senescent T cells, but not all of the immune senescent T cells.
Would you say it's a majority? Is it a major driving factor?
It's donor dependent. It depends on, for instance, the CMV status of the donor. Like if you are CMV positive, you will have a lot of these T-EMRA T cells and so on. So it depends on the insult, on the driving force. But overall, the lack of telomere transfer capacity, which we also discovered a couple of years ago, so the ability of the T cells to get telomeres from the antigen-presenting cells during immune response is definitely a feature of immunosenescence. So even those emery T cells, despite the fact they have slightly longer telomeres than the effector memory T cells, they also lose in part the ability of get telomere transfer. So telomere transfer loss is definitely a feature as regard to telomeres that defines cells that are senescent or on the way to senescence.
Yeah, I want to talk a little bit about telomere transfer, because as you suggested, your work uncovered it sort of a new way. T cells regain longer telomeres, and you call it intercellular telomere transfer. Tell us what's happening there.
So for 40 years, what we knew in the field was that, the dogma was that the T cells, when it gets activated, it turns on an enzyme which is called telomerase, won the Nobel Prize in 2009. Is an enzyme that synthesizing the ends of the chromosomes, and these are the telomeres. So telomeres have to be kept at a certain length, otherwise if they get very short, it triggers senescence cascades in the cells, right? So that is one way of driving senescence. Not the only way, but one way of driving senescence. What we discovered actually is that the story is slightly different in that before even turning on the enzyme telomerase, there is a different pathway for telomere elongation, whereby the naive T cell, so the T cell that has never seen an antigen, when they encounter for the very first time an antigen-presenting cell, that could be a macrophage, that could be a B cell, that could be a dendritic cell. So a cell that has processed the pathogen, has processed the insult, and has presented peptides on the surface to the T cells. So during this recognition process, they form a structure which is called an immune synapse, and this immune synapse serves as the vehicle for the telomere transfer donation. So you have a donor cell, which is the antigen-presenting cell, that donates telomeres in vesicles to the telomere recipient T cells. So the telomeres coming from the DC or the APCs in general, they can be incorporated into the T cells. They get into they get into specific chromosomes of the recipient T cells. So this is very important because the extension of telomeres after the transfer reaction is so much longer than the one that telomerase can do. So telomerase, if you do the math, telomerase can extend telomeres by 100 or 200 base pairs. The average lengthening of the telomere transfer mechanism is 3000 base pairs. So it's much longer. But unlike telomerase that keeps, it's a kind of a buffering mechanism, telomerase. So what it does, it tries to elongate almost all telomeres in the cells and to keep them at a certain length. So with cell division, the cell loses, let's say, 100 base pairs. If telomerase is active, it will put back that 100 base pairs. So meaning that the cells maintain a buffering level of telomere lengthening throughout clonal divisions. Telomere transfer is totally different because it happens before the cells is engaged in the first division. So the T cells will receive the telomere transfer donation before the first cell division. Gets the telomeres, incorporates and elongates the telomeres in his own hands, his own chromosome hands, much longer than telomerase, and specifically to those telomeres in the T cells that are ultra short. This is very important because telomeres, when they're ultra short, they can trigger senescence. So telomere transfer eliminates the ultra short telomeres, allowing thereby to bypass the senescent process before it even manifests itself.
So basically, you've got these antigen-presenting cells, for the most part, that get the telomeric DNA material. They're transferring, so they're sacrificing their own telomere material.
Correct.
For
Correct.
this. So is there, you know,
When you
so
think
what,
about the dichotomy, you have a short-lived cells and a long-lived cells. So
Right.
most of the telomere donor cells are short-lived. You think about the DC, you think about the macrophage, they live weeks or months. You think about the T lymphocyte, they live for decades, right?
Right.
So you understand
So
the
it makes
trait.
sense. So it makes sense. They're basically trying to, they're just trying to keep the T cells living long because they don't really need them.
Absolutely.
For
And
as
most
long.
of them, many of them, they do not even proliferate. You think about the
Mhm.
fully differentiated DCs or macrophages, they barely proliferate, or they do not proliferate. So they function as a factory of telomeres for the T cells.
So at some point, does that, is there a finite period of time in which that happens? I mean, why doesn't this process over time just continue, to mitigate against senescence of T cells?
Yes, that's a good question. It's slightly regulated. So in the first paper we published in HSA Biology, we show the first part of the equation, meaning that you need a naive T cells, let's say, and you need an antigen-presenting cell, but not all the naive T cells can do that. So there is also a metabolic requirement, and we have a follow-up paper under review now in Nature Immunology. There is a follow-up mechanism that makes sure that only the T cells that are correct metabolism in place are worth of a telomere donation. In other words, the DC will not give away their telomeres to any antigen-specific T cell, but only to those T cells that have the correct metabolism in order to be long-lived in the future. So they want to make sure that donation is actually of use to the T cell.
Is that number of T cells that's metabolically capable of doing that,
Yes.
is that dynamic? Does that change?
Yes, absolutely. There is a lot of factors.
So as we age, do fewer and fewer of those T cells allow transfer?
Precisely.
Got it. And so what is it about those T cells, specifically in the metabolism, that, that prevents them from being able to be recipients of this?
I shouldn't be saying this, but I will anticipate. So fatty acid oxidation is the pathway that controls the telomere transfer reaction. I will not go into the details of the metabolic pathway, but only those T cells that have a sufficient level of fatty acid oxidation to trigger very complex lipid cascade that regulates clustering of the T cell receptor in the synapse are capable, in the presence of antigen, to trigger the telomere transfer reaction from the DC. So it's not a blind transfer reaction.
Is there a way to manipulate certain T cells so that they become more receptive?
Absolutely. And that's where ESMAC intervenes. So because ESMAC controls the overall immune senescence, if you target and destroy the ESMAC with our compound of first class DOS, you can reprogram the T cell metabolism and you allow the telomere transfer reaction to occur even at the longer age.
Let's back up so people get that. Your earlier research highlights basically something called P38 MAP kinase. That pathway it's important for T cell aging. Maybe talk about what it is and how it's influencing.
So if you go to textbooks, you will see that MAP kinase cascades have been highly studied, like in the 80s. They have all these amazing works of the transphosphorylation cascades. So when I started, I was younger myself and I was studying myself the P38 pathway. We started from the observations that P38 was spontaneously active in senescent T cells, but no one had the idea of looking for a simple question like what are the upstream triggers that are responsible for the activation of P38. So when we investigated upstream of the kinase, we then discovered that all the classical cross-phosphorylation cascades are absent from these cells. And then we found out that it was this different complex responsible for the activation of P38, which is the paper you refer to, but also of ERK and JNK. So altogether, they reside all together in a complex with senescence and AMPK. It's a gigantic complex that sits in the reticulum endoplasmaticum, so in the ER, and it controls through different MAP kinases the senescence phenotype of the immune cells completely and totally independently from the transphosphorylation MAP kinase cascade that we described. So you need, so if you go and target specifically this complex processor inhibitor that is specific for the ESMAC, you can unlock the beauty of the rejuvenation phenotype. And these things happen very quickly. They happen overnight. So overnight you can, get back in time of 20 years, 25 years, cells that are completely senescent, they go back to stem state.
So basically, again, you've got cestrins, which form complexes with MAP kinase, right?
And AMPK. And
AMPK. And under conditions of stress, or I guess aging?
Yes, DNA damage, nutrient deprivation, oxidative stress, and so on.
And so they intensify signals that ultimately drive T cells towards senescence.
Yes.
Is that
absolutely.
correct? And then so do we know specifically what triggers the cestrins and the MAP kinase and the MPK, that complex? Do we know what triggers that?
Yeah, the DNA damage that activates P53. So the cestrins, in particular, cestrin 1 and 2, they are transcribed by the P53. So all the
Mhm.
pathways that converge to activate P53 to turn on the sessions. But it's very important to also understand that previous work, which you've seen work from David Sabatini, they described the different function of sessions than instead anti-aging. It has nothing to do with the ESMAC we're describing, simply because in those circumstances, sessions are not sitting in the ESMAC, but are instead bound to the GATOR and mTOR complex. So in that complex, they have a totally different function. So when sessions are instead found in terminally differentiating T cells in the ESMAC, they have pro-aging function. So it's very important pharmacologically, if you want to block sessions for rejuvenation, it's very important that you specifically target sessions when they are only in the ESMAC. And this is what we have done with the DOS, which is the first technology in the world that specifically and only destroy and target and interfere with the action of sessions only when they are in the ESMAC, sparing sessions when they're in the mTOR and GATOR. So sparing the good anti-aging function of sessions and completely only focusing on the function of sessions in the ESMAC, which is pro-aging.
And just so that people understand, again, making sure that we don't get ahead of ourselves. So you
Sure.
guys developed these compounds, which you're talking about DOS, which are disruptors of cestrin MAP kinase complex.
Mm-hmm.
And
Absolutely.
well, you also mentioned that part of what those disruptors are doing is preventing these T cells from becoming metabolically such that they can no longer become recipients
They
of
restore the correct metabolism, because if you look
yeah.
into also the literature, it is well known, it's not secret, that T cells, when they are senescent, they are extremely glycolytic. So what the DOS technology does, among all the amazing things it does, but metabolically speaking, it actually shifts again the equilibrium back to fatty acid oxidation, which is the correct program to allow the transfer reaction of telomeres.
But once a cell is T cell senescent, you can't reverse it from there, can you?
Yes, I can.
Oh, you
Yes, I
can. You
can.
can. So
Yeah.
this isn't just
Overnight.
this isn't prevention. This is
It's
actually
not prevention. This is not
reversal.
prevention. For prevention, we have a different class of drugs that are called NOS, negators of ESMAC. They were a different paper now in submission to immunity. So, because in that case, you need to break into the mechanism responsible for ESMAC generation, which is not known, and we have not disclosed it as yet. So we're talking about T cells that are already senescent or on the way to senescence, that have already formed ESMACs inside their cytoplasm in proximity of the nuclei, and you want to target and destroy those complexes. If you want to prevent the aging process, it's an amazing question from you. Thank you for the question. If you want to prevent the aging process instead, you need to prevent the genesis of the ESMAC. So it's a completely different pathway.
So, how much of this research is in vitro? How much of it is in the lab? How much in cells versus how much of this have you seen in animal studies?
Yeah, it's the same. We've done side to side. So the beauty of it is that the system is mechanistically evolutionarily conserved. So you can study it into primary human T cells in vitro in the lab, and you can study it into the animals in vivo, and you can time the reaction. We know also the correspondence on the process. So processes, I would say, like an example, processes that in the lab would happen in 4 hours. In vivo, the same process takes 2 days. So we know, of course, the timing is slightly different, but the effect, biologically speaking, is identical.
And what kind of phenotypic differences? Like what do you see in differences in these animals who've been treated? Do you carry out long enough experiments to see, physically if how they're different from?
Yeah, they are very different. So we have a follow-up paper of the telomere transfer story where actually we show that when you act on these animals and you restore the telomere transfer reaction and so on, we describe a process that whereby these T cells are capable to elaborate the telomere transfer reaction. There is a leftover of the reaction, and they release flows of telomeres in the circulation. So these flows of telomeres in circulation, they go everywhere systemically. They rejuvenate the appearance of the animals, they extend the lifespan of the animals. So think about a mouse lab lifetime lifespan is about 2 years or 2.5 years. We can push it to 4.5 to 5 years with a single injection of the DOS.
In a rat, you said? In rats?
In a mouse, yes.
In a mouse. Got
Yes.
it. Got it.
So they change in
Interesting.
the appearance, they change also the coat is more glowing and so on, the lifespan, the resist to infection even without vaccination. The immunology effects are in a preprint already, and the systemic ones instead, they are under review. They're both under review, but they're two different papers.
Yeah. Do you, in terms of human use here, how realistic is it for us to see various kinds of T cell rejuvenation therapies with something like these DOS compounds in practice?
We're starting after the summer at UCL. We have set up a translational lab for phase 1 studies, and we are starting with, of course, safety, because it is the first time in the world that the ESMAC is blocked in humans. And something very exciting that I feel to add, which we discovered in part serendipitously, in part because we looked into it, is that part of this mechanism of T cell rejuvenation offer the first cure to HIV. So we're launching a study. Yes, so we can get rid very easily and very quickly of the virus, which is integrated into T cells from infected patients. And we're very excited because, with 1 or 2 or 3 injections of this technology, we can completely remove the integrated HIV.
And you've seen that happen in mice already? So,
Yes.
interesting.
In T cells from people, from patients in vitro, and injecting the patient's T cells into the animals, and in mouse models of the disease.
Yeah. What is there? So I guess from your standpoint of you're doing safety tests right now for something like this to potentially become, would it come out as a drug, as a therapeutic for a specific disease? Is that how it
Well,
would first come out?
that is a tricky question, because as you know, longevity from a regulatory standpoint, unfortunately, and aging is not seen as a disease as yet. So what we do, obviously, we try to target an age-ready diseases, but we are very much clear that this is a platform that can contribute to cure all sorts of age-ready diseases.
Sure.
Because the more we study and the more we understand, as I said at the beginning of this interview, that the age of the T cell, we need to redefine, we need to think again about what is the function of the immune system. The function of the immune system is not just responding to infections or taking care of cancer cells. The function of an healthy immune system is taking care of the entire body. So all the diseases that appear with age, they are somehow connected with immune dysfunction. So we believe that by rejuvenating the immune system, we can offer a new solution for a plethora of age-related diseases. So we will start with vaccination. Vaccination is an easy one. The vaccines, unfortunately, do not work very nicely in older people. Nowadays, after COVID, we are seeing actually a lot of circumstances in most of donors, even at 25 or 30 years of age, the immune system is highly damaged. And therefore, there is an active interest in trying to rejuvenate the immune system, even at younger age, that before we didn't think it usable. And instead, is actually, it offering us the opportunity to test principle of this technology, even in people who are 30 or above. So we'll start from people who are probably 40 onwards in the context of vaccination and HIV. But we know that it works without vaccinations, so we can offer prophylactic protection also if you don't get a vaccine, just giving the immune rejuvenating product that we created.
And by the way, these trials that you're doing, are they being done in Europe or in the US?
In the UK. In
the UK. Okay, got it. I'm just kind of curious in terms of like how that generally, I mean, in the US, everything takes a long time. I'm assuming in the UK, it's going to take just, it's not going to be any faster to move things along.
Right. Exactly. Well,
Yeah.
we hope to be in the market in 5 years, at least
Yeah.
for the first application. So that is what we're working for.
While we wait in those next 5 years, just, I guess, last question for interest for people. Are there any other interventions of people, supplements, things like that you think have some potential to maintain a healthier immune system right now?
Well, unfortunately, there is not much. Of course, there is a good exercising. Obviously, there is all the things we already know. But this is really, the first the first way pharmacologically to reprogram our cells when they got senescent. And we never
Yeah.
thought about this before. So we were thinking about exhaustion and all other states that regulate immune dysfunction, but senescence has been largely overlooked.
Anyway, fascinating stuff. I really do appreciate your time today. Where can we learn more about your company and your work?
Yeah, you can type in centicell.life and understanding all this pharmacology I described. We also launched a side project which is very fun from rejuvenating cosmetic appearance, which is called the Infinum. So it's actually that product is on the market, and it's based on some of the I would say concept and know-how we have developed, obviously for cosmetic uses, but it feeds into that. That is probably what is closest to what the drug will be once injected. Obviously, it's a cosmetic, but the
Right.
principles are similar and triggers telomere transfer in people.
And what was that? What was that product called?
The Infinum.
Infinum. Okay.
It's
got it.
just a peptide cream, but triggers telomere transfer in human skin.
Yeah. Fantastic. Thank you so much, Alessio. I'd love to have you on again at some time.
Thank you.
Thanks for listening. A quick reminder that while I am in fact a surgeon, nothing I say should be construed as medical advice. Now make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.